1 minute. Okay. Now I will talk about the oogenesis. So the oogenesis is the production, the process by which there will be a production of female gametes or of oocytes and ova. And the oogenesis occurs in the ovaries. Those are the ovaries. So here we have a female reproductive system showing the ovaries, the fallopian tubes, the uterus, and the vagina. So the oogenesis occurs in the ovaries, and the oogenesis is a discontinuous process that occurs monthly. So it's unlike the spermatogenesis. We have said, or we have seen that the spermatogenesis is a continuous process from puberty until senescence. It can keep on happening. And also, we can always have cells which will engage in spermatogenesis in a continuous way. However, during the oogenesis, or the oogenesis is a discontinuous process. This process will not be maintained all over a female life. It will stop at the menopause. And also there are many arrests during the process of oogenesis. As we are going to see, the meiosis during oogenesis will be arrested first before birth and will resume after puberty, and not in all of the cells. It will resume only in a pool of cells, in a few cells at each menstrual cycle. So the process of oogenesis will, after puberty, will take place at each menstrual cycle, and only in a few cells, not in all of the cells. And also we will have arrests in the meiosis, which is unlike the spermatogenesis, where the meiosis is not arrested. When it starts in the primary spermatocytes, it continues until the end without any arrest, and it is not disrupted, and the process of generating of gametes is not disrupted also, like in the females, as we are going to see. So in the females also, it does not occur all over the life of a female. It will stop at the age of 50 or 55 at the menopause. So what are the steps of the oogenesis? During the oogenesis, we have a proliferation phase, a growth phase, and also a differentiation or a maturation phase. So during the proliferation phase, here we have before birth, in the fetal life, during the embryogenesis, we will have a development of primordial germ cells in the female. And those primordial germ cells will divide in the female ovaries. Those primordial germ cells will divide by mitosis to give oogonia. So the singular is oogonium. So each primordial germ cells will divide to give oogonium, an oogonium or oogonia. So here we will have oogonia generated from the division of primordial germ cells, and those oogonia will divide also, will divide extensively before birth by mitosis to give one identical oogonium to the initial one, to conserve this pool of oogonia, and to give a more mature another cell which will engage in meiosis, which will be the primary oocyte. Okay, so during the fetal life, we will have those primordial germ cells which will divide to give oogonia, and those oogonia will divide also extensively. So during this fetal life, from between the 2nd and the 7th month, we will have the proliferation of those oogonia that will take place. And by the end of the 7th month, the fetal ovary will contain several millions of oogonia. Okay, so this is the proliferation phase. The proliferation phase, we have the proliferation of the primordial germ cells to give oogonia, and then the oogonia, which will proliferate extensively to generate several millions of oogonia, a stock of oogonia. Now, those oogonia, a lot of them, so approximately half of them will be destroyed or will die before birth. Okay, so we are still talking before birth. Okay, half of the generated oogonia will die by apoptosis or atresia. So the mechanism of the cell death is called apoptosis or atresia. And half of them will remain. So the half of the half of the remaining oogonia will enter the meiosis to become primary oocytes, and this is the growth. So this happens during the growth phase. Okay, so during the proliferation phase, we only have the primordial germ cells which divide to give oogonia, and the oogonia that will divide by mitosis and that will generate a stock of oogonia. This will happen during the proliferation phase before birth. Now, also, I should say that the growth phase occurs also entirely before birth. So those two phases, the proliferation and the growth phase, occur before birth. Now, during the proliferation phase, the oogonia, as you said, they proliferate. They are diploid cells. They are enlarged, spherical. They are round-shaped cells, spherical, and they are not differentiated. So they are stem cells. They are the most immature cells at the origin of the other germ cells. Okay, and also, as we have explained during the spermatogenesis, the cells dividing or coming from one oogonia, as we have said, there will be many divisions of the oogonia. So the cells dividing and coming from one oogonium will be connected to each other by cytoplasmic bridges that will allow them to, or that will allow their synchronous differentiation. So they will divide at the same time, and whenever we will have the atresia, and they will die, some of them will die, and the others will enter the meiosis at the same time. Now, so this will happen during the proliferation phase. During the growth phase, we have the atresia of the oogonia. The remaining oogonia will enter the meiosis. Once they enter the meiosis, we call them primary oocytes. Okay, we don't call them anymore oogonia, but we call them primary oocytes. And those primary oocytes, they will start the meiosis, and they will progress in it until the diplotin of the prophase 1. If you remember, we talked last session that the oocytes will be blocked or will be arrested at the diplotin of prophase 1. So here, whenever those primary oocytes will reach the diplotin of the prophase 1, they will be arrested at this stage. Okay, they will no more continue the meiosis, and this meiosis will be arrested here. they will undergo a growth phase. They will increase in size during the diprotein stage, and the cytoplasmic bridges, they will be disrupted between the cells. Okay. So they will remain here. The cells will remain. We have generate the oogonia. Some of them during the growth phase, half of the oogonia will die, the other half will enter the meiosis. They will be called primary oocytes. Okay, those primary oocytes will enter the meiosis and will be arrested at the diprotein of the meiosis 1. The cytoplasmic bridges between those primary oocytes will be disrupted, and they will enter a growth phase. They will increase in size, as we have said, that during the diprotein stage, they will produce reserves of protein. They will produce messenger RNA and proteins that will constitute a reserve for the later stages of oocyte development and for the fertilization and the zygote division. Okay, so they will increase in size, and this is and they will be maintained blocked in this stage until puberty. So once a female is born, it will be born with a pool of primary oocytes blocked at the diprotein stage. Okay, and this meiosis will remain arrested until puberty, and it can, in fact, at puberty, not all of them will resume the meiosis. At each menstrual cycle, only few of those primary oocytes will resume their meiosis, and the other will be maintained arrested in their meiosis. So the arrest of the meiosis of this in the diprotein of meiosis 1 can be maintained for very long periods of time, and one oocyte can wait until the menopause or just before the menopause to be for the meiosis to be activated. Okay, it depends if at this menstrual cycle it will be chosen, let's say, to continue the meiosis or not. So the primary oogonia will remain arrested at this stage until puberty and until long periods of time. Okay, so here we can see that when a female is born, she will be born with a pool of primary oocytes arrested in meiosis. They will not have a pool of stem cells of oogonia, because those oogonia are transformed into primary oocytes, became primary oocytes. And at each menstrual cycle, few of those primary oocytes will resume their meiosis, and the others will wait for another menstrual cycle. And that's why at the menopause, the age of menopause, it is the age where she won't have any more primary oocytes to be engaged in a new cycle to resume their meiosis. All of them will be used or will get activated and ovulated or will die. So a female after the menopause, she won't have any more primary oocytes. Okay, that's why the reproductive life of a female will arrest at a certain time. However, in the males, we always have generation, as we said, of sperm cells during a male lifetime. They always have spermatogenesis. However, in the female, no. She will be born with a certain number of cells, which will continue the oogenesis. Okay, so during the growth phase, we will have apoptosis, atresia, and there will be destruction of approximately half the number of oogonia. And for the remaining oogonia, they will enter the first meiosis to give primary oocytes. The primary oocytes will progress in meiosis until diprotein stage of prophase 1. the meiosis is arrested in diprotein stage of prophase 1, and the primary oocytes will remain blocked in the diprotein of prophase 1 until puberty. Then the intracellular cytoplasmic bridges between the primary oocyte will be disrupted, and the primary oogonia will enter the growth phase. So they will increase in size and will be synthesizing their reserves for later stages. Now, those two steps occurs only in before birth. Now, after birth, during the childhood, nothing. We don't have any development, any of the oogenesis. We don't have anything. It's only after puberty and at each menstrual cycle that a primary oocyte, which was formed before birth, the primary oocyte will resume the meiosis, and so it can continue its meiosis. So now after puberty, let's say one primary oocyte can resume the meiosis. It will divide to give 2 cells at the end of the meiosis 1. It will give one secondary oocyte and another small cell called the first polar body. So the division in the primary oocyte is not equal. The cytokinesis is not in the middle, okay, is not equal. So that's why we don't have equal 2 cells equal in size. We have one big cells and one smaller cells, which is the first polar body. And this big cell is the secondary oocyte. It is a haploid cell. This haploid cell, this secondary oocyte, will engage in the meiosis 2, so will directly enter in the meiosis 2, but however, it will get arrested again at the metaphase of meiosis 2. Okay, so this secondary oocyte will get arrested at the, will start the meiosis 2, but will get arrested at the metaphase of meiosis 2, and it will be ovulated as an arrested oocyte in the meiosis 2. So it will be ovulated while arrested in meiosis 2. You know, when it is ovulated, it will be expulsed from the ovary and it will go to the fallopian tube. And here in the fallopian tube, we will have the fertilization process. So if this secondary oocyte is fertilized, if it will be fertilized, the meiosis 2 will resume, can continue, and it can be completed. It will be completed to give 2 other cells. So this secondary oocyte, if we have fertilization, it can achieve its meiosis 2 to give 2 cells. Also, the division is not equal, so we will have one big cell, which will be the ovum, and another small cell, which is a second polar body. Okay. So if this after ovulation, we have this secondary oocyte or this oocyte, if it will not get fertilized, the cell will remain blocked at metaphase 2 until it will die, and it will be until it will die. Okay, so it will the meiosis 2 or the meiosis in the oocytes will only be achieved if there is fertilization. If there is no fertilization, the meiosis will not be achieved. Okay, so once we will have a fertilization, we can have the cells that were arrested at the metaphase 2 can resume the meiosis, and we will have an ovum, which is a haploid cell containing one chromatid for each chromosome, and we will have a so as a result of the fertilization, we will have the fusion of the 2 nuclei of the cells, and then we will have the a fertilized egg. Okay, but the fertilization is essential and is a condition to complete the meiosis and then the maturation of the oocytes. If we don't have fertilization, the maturation cannot be accomplished. As I said, when I talk maturation, I'm talking about meiosis. So when I say the meiosis, we mean also this is the maturation phase. So the maturation phase starts from puberty till menopause, and it is characterized by the resumption of meiosis, because as you know, before birth, we had this meiosis arrested at the diprotein 1. So after puberty, during the maturation phase, we have the resumption of meiosis, and the primary oocytes initiate the maturation phase. So some primary oocytes will start or will resume the meiosis, will initiate the maturation phase, but not all of them at the same time. At each menstrual cycle, only few of them will get matured, will continue or will resume the meiosis, and not all of them at the same time. So what are the steps of the maturation? At each menstrual cycle, a few primary follicles start to develop. They will resume their meiosis 1. You know the difference between follicle and oocyte. So here we will see this later, but the oocyte during before birth will be surrounded by follicular cells, and we will call it a follicle. We will have ovarian follicles, okay? But the oocyte will be inside of the follicle. So at each menstrual cycle, few primary follicles will start to develop, and they will resume their meiosis 1. So they will start to mature, and they can resume their meiosis 1. After the first meiotic division, we will obtain 2 haploid cells. One of them is the secondary oocyte, which is large, and the other one is a small cell called the first polar body. And after the first meiotic division, after it is completed, then start the second meiotic division, where the secondary oocyte will be arrested. So this secondary oocyte will start the second meiotic division. It will be arrested at the metaphase 2 until the fertilization. So it will be ovulated while arrested at the metaphase 2. So the secondary oocyte is shed from the ovary and enters the fallopian tube as secondary oocyte arrested in metaphase 2. And after fertilization, the secondary oocyte completes the meiosis and divides. After the fertilization, it can complete the meiosis and divide into 2 other cells. One big cell, which is a large oocyte or ovum, and another small cell, which is the second polar body. So the maturation or will be accomplished. So the accomplishment of the maturation or of the meiosis is conditioned by the fertilization. If there is no fertilization, there will not be accomplishment of the meiosis. And as I said, the cell will keep on being arrested at metaphase 2, and then it will die. Okay. Here we can review a little bit the different steps of oogenesis. We have during the fetal life the oogonia, which will divide extensively by mitosis, and at a certain moment, those oogonia, as we said, some of them will die, a lot of them will die, and the remaining one will enter the meiosis, okay, to give primary spermatocytes. One of those cells enter the meiosis, we call them primary, they will become primary oocytes. And the primary oocytes, they will start the meiosis, but they will be blocked at the diprotein of the prophase 1. We have cells connected to each other by cytoplasmic bridges. Those cytoplasmic bridges will be disrupted, and when they are arrested at the diplotin stage. They will undergo a growth phase where they will increase in size because they will synthesize messenger RNA, translational RNAs, proteins, so reserves that will be used later by the oocyte. Okay, so this is all. Here we have before birth the proliferation and the growth phase. In the childhood, nothing is happening. After puberty, after birth and after puberty, the oogenesis continues, and some of the primary oocytes at each menstrual cycle, few of the primary oocytes will resume their meiosis to give one secondary oocyte and another polar body. So those are haploid cells having two chromatids for each chromosome. And the secondary oocyte will engage in the meiosis 2, will start the meiosis 2. However, it will be arrested at the metaphase 2 of this meiosis and will be ovulated and arrested at metaphase 2. If there is fertilization, this secondary oocyte can continue, can achieve the meiosis and give two cells, one ovum and one second polar body, which are two haploid cells having one set of chromosomes and one chromatid per chromosome. Okay, so before here, the primary oocyte is a diploid cell containing two chromatid per chromosome, and also are the oogonia. Here also we have another figure. Before birth, we have a primordial germ cells which will divide to give oogonia. The oogonia will divide several times, and at certain moment they will become primary. They will enter meiosis, and we call them now primary oocytes. And those primary oocytes will get arrested at prophase 1 of meiosis, and at the diplotin of prophase 1, and they will increase in size. This is the growth phase. Now, at each menstrual, during the childhood, nothing will happen. After birth, and it's only after puberty. So the puberty is a sexual maturity. So it's only after puberty that the oogenesis can be achieved. And so at each menstrual cycle, one primary oocyte will be, not one, but few of them will resume the meiosis to give secondary oocyte here. And the secondary oocyte will enter the meiosis 2, but will be blocked in the metaphase 2. When we will have the ovulation, it will be ovulated as secondary oocyte blocked at metaphase 2. If there is fertilization, the secondary oocyte will give an ovum and a second polar body. If there is no fertilization, it will be kept, it will remain blocked at metaphase 2. Okay, this is regarding the oogenesis. Now I will talk about the similarities between the spermatogenesis and the oogenesis. As we have said, the gametogenesis for both of them, in both the spermatogenesis and the oogenesis, we have a proliferation phase at the beginning. So the gametogenesis in both cases will start with a proliferation phase where we have diploid germ cells, the spermatogonia and the oogonia, which will divide by mitosis. Okay, as we have seen here, the oogonia, or here the oogonia, and in the male, the spermatogonia, that will divide to give primary spermatocytes, and in the females to give primary oocytes. Okay, so we have a proliferation phase common for both of those processes. And when we say proliferation, we say mitosis, division by mitosis. Okay, and in both cases also, the mitosis will cease at certain moment, because during spermatogenesis, The mitosis will get arrested before birth. As we have said, the spermatogonia will divide, or spermatogonial stem cell will divide before birth. After birth, we don't have anything important happening, and after puberty, they start to divide again. Okay, so we have the mitotic divisions that will cease at a certain moment during the spermatogenesis and during the oogenesis. As we have said, the oogonia will stop dividing at certain moment and they will become primary oocytes. Okay, so also here the mitosis will cease. But the difference is that during the spermatogenesis, we have mitosis again and proliferation again after puberty. So the spermatogonial cells can divide again after puberty to engage in new, to produce new spermatogonia and other cells. However, in oogenesis, we will not have mitosis anymore. It's only the primary oocytes that will continue their meiosis, okay? But we don't have any more oogonia that will divide to give primary oocytes. This step is the proliferation phase is reserved for the phase before birth in the females. And since we have meiosis in both of spermatogenesis and oogenesis, and as we have said during the last session, always before the meiosis, the start of the meiosis, we have a DNA duplication that takes place during the interphase before the beginning of the meiosis. And of course, we mean the meiosis 1. Okay, so we have a DNA duplication that takes place during the interphase before the start of the meiosis. And this is common for both of those, for both of them. The germ cells become primary spermatocytes and primary oocytes. Also, the germ cells in the male will become primary spermatocyte. The spermatogonia, as we have seen, they will become primary spermatocytes, and the oogonia, which are the germ cells in the female, will become primary oocytes. And both of them, the spermatogenesis, and during both of them, the spermatogenesis and the oogenesis, we have a growth phase. So the primary spermatocytes, as we have said, here the type, the spermatogonia will enter a growth phase and will become primary spermatocytes. Okay, and also in the female we have a growth phase where the primary oocyte will increase in size once they are in the diprotein of the prophase 1. So during growth phase, primary oocytes and primary oocytes increase in volume. So the growth phase means that we have an increase in volume in the cells, and this happens in both the primary spermatocytes and the primary oocytes. But the increase in the size of primary oocytes is much more important than the increase in the size of the primary spermatocytes. So the cytoplasmic growth in the oocytes is more important. And in both of them, we have the meiosis process. Okay, so the meiosis starts. It is the same process for spermatogenesis and oogenesis. Those are the similarities. Now, what are the differences between both of them? We have a difference regarding the duration of the process. So it is the time required to transform a gonia or an oogonium into, agonia. It can be a spermatogonium or a oogonium, depending if we are talking of spermatogenesis or oogenesis, to transform germ cells, stem germ cells, into mature gamete. So in the spermatogenesis, or the spermatogenesis proceed more rapidly than the oogenesis, it lasts several weeks. As we have said, the spermatogenesis needs 74 days to be accomplished. So we need 74 days to pass from spermatogonia to sperm cells, to mature sperm cells. However, the oogenesis, the cycle or the process can endure for all the sexual life of the female, from before birth until just before the menopause. Okay, it can be very long depending on when the primary oocyte will resume its meiosis. Another difference is regarding the proliferative periods. So during the spermatogenesis lasts from puberty to senescence. Okay, the proliferation in spermatogenesis can happen. It can take place from puberty till senescence. We always have spermatogonia that can proliferate, divide, and then give primary spermatocytes, etc. Okay. However, in the oogenesis, the proliferative phase is only present during the fetal life, where the oogonia will divide by mitosis to give other oogonia, and then they will become primary oocytes. So this proliferation is restricted only to the fetal life. Okay, and it is not maintained all the long of a female life. Okay, and during the oogenesis, we have only a limited number of oocytes that undergoes a growth and maturation phases during cycles that follow from puberty till menopause. So here we only have limit, as we said, at each menstrual cycle, only few cells will resume the meiosis. And to give gametes, okay, not all of them. Now, we have difference regarding the importance of the growth phase, as I have precised earlier, that the increase in the cytoplasmic volume of the spermatocytes 1 is reduced compared to the increase in the size of oocytes. So the spermatocytes 1 volume increases a little compared to compared also to spermatogonia. So here, during the growth phase, we should have an increase in the size of primary spermatocytes compared to the spermatogonia. So the increase in the size of primary spermatocytes is little compared to the spermatogonia. However, during the oogenesis, the growth, we have a more important growth of the primary oocytes. The primary oocytes will increase more consequently in size, because they will increase and accumulate reserve. They will increase the number of reserves and inside of the cytoplasm. The meiosis in the spermatogenesis is initiated continuously in the males in a mitotically dividing stem cell population. So during the spermatogenesis, as we have seen, the meiosis is continuously initiated. Since we have spermatogonia, they are dividing, they give primary spermatocytes, and those primary spermatocyte can enter the meiosis without any problem. So we have a continuous a meiosis that can happen continuously during the life of a male. And it proceeds continuously without cell cycle arrest. Also, the meiosis, when it happens in the primary and secondary spermatocytes, it is not arrested. It's not arrested at the diplotene stage like in the female, and it's not arrested at the metaphase 2 like in the secondary oocytes. Okay, so there is no cell cycle arrest during the meiosis during the spermatogenesis. Okay. However, during the oogenesis, the meiosis is initiated once during the lifetime, and it is initiated also during the fetal life. After puberty, it will only resume, and it will not be initiated again. So after puberty, there is no cells that will start the meiosis. No, only the meiosis will resume in the cells which were blocked or arrested in their meiosis before birth. So during the oogenesis, the meiosis is initiated once before birth. It is only here before birth that we have the meiosis that occurs in the primary oocytes, and that's all. After this, we will have those cells that will continue their unachieved meiosis, but there is no other cells that will start or will enter the meiosis again, okay? So in the female, during oogenesis, the meiosis is initiated once during the fetal life and in a limited population of cells. We don't have a pool of cells that always can always enter the meiosis. And the meiosis is arrested at the first meiotic prophase and reinitiated in smaller population of cells. As you know, the meiosis also is arrested. Is not continuous here like in the spermatocytes. It will be arrested at the prophase of diprotein 1 in the primary oocytes, and then it will be arrested again at the metaphase 2 in the secondary oocytes. So we have arrests in the meiosis. Now, in the male, we have a difference regarding the place in the meiosis in the process. So when does it happen, before or after the differentiation of the cells? During the spermatogenesis, the meiosis precedes the differentiation of the spermatozoa. As you see, we have the meiosis here, and then at the end of the meiosis, we have spermatids, which will undergo a differentiation. Okay, so the meiosis in the males happens before the differentiation. However, in the females, during oogenesis, now whenever they talk here about in the females, when they talk about the differentiation, they talk about the growth phase. Okay. It is different than what they mean by differentiation in the male. Here in the male, we mean by differentiation, the differentiation from spermatids to spermatozoa. And during oogenesis, they mean the growth phase. When they say differentiation, they mean the growth phase. So during oogenesis, the growth phase or the differentiation of the oocyte occurs while the cell is arrested in meiosis. Okay, here the growth phase, once we have those primary oocytes, they start the meiosis. They are blocked at the diprotein stage, and then we have the growth phase. So the growth phase or the differentiation occurs while those cells are engaged in the meiosis. Okay, in the male, no, it starts, the meiosis precedes the differentiation. Here in the females or in the oogenesis, the growth and differentiation occur while the cell nucleus or the cell remain in a prolonged mitotic prophase. Now, the differentiation in spermatogenesis occurs while the cells are haploid after meiosis. So here, as you can see, when we have the differentiation, the cells are haploid cells, the spermatids are haploid, and they undergo, or they will be the subject of the differentiation. Okay, so they are haploid when they start the differentiation. In the females, when we have the growth phase, the cells are diploid. They are still blocked at the meiosis 1, so that the growth phase occur while they are blocked at the diplotin of prophase 1, so they are diploid cells. Okay, so the differentiation in oogenesis occurs while while the cells are diploid blocked in their first meiotic prophase. And we have a difference regarding the results of the meiosis. During the spermatogenesis, we obtain 4 gametes from one primary spermatocyte. So each primary spermatocyte will produce 4 functional gametes. However, during the oogenesis, we will obtain only one ovum. Okay, we will obtain one ovum and one polar body. So we will obtain mainly one functional cell, one gamete. Okay. As a result of the oogenesis, we will obtain one gamete, which is a functional reproductive cell. However, during the spermatogenesis, we obtain 4 functional reproductive cells, 4 functional gametes. Okay, do you have any questions?
No.
Okay. So this is all regarding this chapter. I hope it was clear.
Yes, it is.
Okay, next
Thank
time.
you, doctor.
Yes, welcome. Next time we'll start another chapter, okay? We'll start the chapter 2.